session, and also a very warm welcome to all the distinguished speakers for this session. I will ask Dr. Amita Mukherjee to introduce the speaker for this session.
Good evening. This is the second technical session post-lunch. Welcome all of you in the second technical session. There will be 3 talks in this technical session. To start with Dr. Sandhya Devella's talk. Let me introduce her. She has completed her PhD from IIT Chennai or IIT Madras, that is known, in 1998 in area of analytical chemistry. From 2000 to 2003, she was a postdoctoral research fellow for the chemistry department, University of Aberdeen. And her work involved understanding the aquatic arsenic species found to the lipid, a special breed of seaweed eating ship. Then from 2003 to 2015, Sandhya worked at Marine Scotland Science, a directorate of the Scottish government, as an environmental chemist. She worked on monitoring and assessment of organic and inorganic contaminants from the marine environment and work related to regulation also. 2015 onwards, she has been working as a scientist in James Hutton Institute, where she is involved in research and commercial works. And she is the section head for the commercial organics now. She has started working in the area of microplastics, where she is co-supervising a PhD student and was successful in obtaining a Shipton grant also. She is a selected member of Food Standard Agency Registry of Specialists in the area of analytical chemistry, method of analysis, and organic and inorganic contaminants and food. So I welcome Dr. Sandhya on behalf of the organizing committee. Please share your views with us. Thank you.
Thank you. Can you see my screen?
No, not yet. Please share your screen.
No. Dr. Mridula, please stop sharing screen. Yeah, now we can see.
Now it's fine.
So thank you for the introduction. I would like to discuss some of the challenges we faced in the separation of microplastics from terrestrial soils. This work within the Institute involved a few of us, Dr. Zulan Zhang and Pat Cooper, who are with the Organic Analytical section, Dr. Jean Robertson and Dr. Reza Hege with the FTIR section, and Stuart Ramage, who's recently joined for PhD. Before I move on, I would like to mention that it's only recently that within the Institute, we started moving into microplastics research. And the outline of my presentation will be a brief introduction of who we are as an institute and why are we interested in microplastics analysis, followed by what work have we done so far. This is only preliminary work that I'll be presenting the challenges, some of the challenges we face and possible solutions going forward. So James Hutton Institute was formed in 2011 by merging what was formerly the Macaulay Land Use Research Institute at Aberdeen and the Scottish Crop Research Institute at Dundee. We also have 2 research farms currently associated with the Institute to help research. Until October last year, we had the Hartwood Research Facility, which is no longer being leased by the Institute. The Institute undertakes diverse areas of research. I've just shown some of them. And one of the important components of the research at the Institute is soils. And soils are considered as a finite and valuable resource that need to be safeguarded for a sustainable for maintaining diversity, fertility, and productivity. And James Hutton Institute is regarded as one of the largest skill centers in Europe for soils research. So why are we interested in soils analysis for microplastics? The Institute is host to a National Soils Archive, which is a collection of over 43,000 samples collected across Scotland, and it's well preserved over a 2.5 kilometer shelving. We have samples way back from 1934, and one of the important components of samples into this archive is from the National Soils Inventory of Scotland, where soils were collected over every 10 kilometer grid, and later on between 2007 and 2010 over 20 kilometer grid. Each soil has about 100 attributes associated with it, where the sample has been analyzed for chemical and physical properties and also land characteristics. And these samples have been used for several long-term and nationally significant experiments. We would like to analyze these soil samples for microplastics to look at the prevalence of microplastics in Scottish soils. We also have samples from the Hartwood Research Facility, which were samples collected for a previous long-term sludge experiment. So this experiment was to test the long-term effects of sludge treatment on agricultural soils. We have those soil samples available from 1998 to 2013. So we've got two different time periods to analyze the samples, and sludge is now considered one of the main components of microplastics pollution into the terrestrial soil environment. Looking at the distribution of microplastic publications in various environments, this is a 2018 paper, only about 4% of soils of the publications are devoted to the soil environment, compared to the marine or freshwater environment and the biological samples. Although there is increasing evidence of microplastic prevalence in the soils, and there is evidence that they are harmful to the living organisms. One of the most common methods of separation of microplastics from soils has been the density separation method. However, soils can be complex mixtures, because not just variations in the clay, salt, and sand content, but also because of the presence of different levels of decomposed products or organic matter, which can have densities similar to the microplastics, and they can be a microscopic matter which can interfere with the density separation. The picture on the right shows I've given the reference below. Shows the amount of papers that have been devoted so far to the method development part of the microplastics analysis. The small proportion here, wheat colored ones, are the number of papers devoted to this area of method development, which is important in the microplastics area of research. So as my colleague Zulin and his team were involved in developing a density separation method for microplastics. It's got 4 different steps. The first one, this is just an overview. It's the density separation. We take 40 grams of air-dried and less than 2 millimeters sieved soil samples and subjected to density separation using zinc bromide. The next step is the interference reduction. We are using hydrogen peroxide, which is a mild digestion step. We can also use the Fenton's reagent, which is slightly more powerful than the peroxide, but I do not think it will cause a much more reduction than the hydrogen peroxide, because what we're seeing is the peroxide step is only bleaching the interference rather than causing a reduction in the non-microplastic matter. The next step is the enumeration step, where the microplastics are counted using an optical microscope. I was involved in a Nile red dye staining method, not really adapting a Nile red dye staining method using fluorescence microscope. It does have positive interferences, but recently there was a paper published where a co-staining approach was proposed to overcome this problem. And I think together we can use the optical and fluorescence microscopy complementary to each other to improve the to reduce the margin of error. And this step gives us important information about the not just the number of microplastics, but the shape, and the size, which are important factors for because the effects vary depending upon the shape and size in any given environment. And finally, the microplastics are identified by the ATR, FTR microscopy. And so in this presentation, I will be focusing mainly on the first two steps to improve our existing methodology. We have applied the method we have so far on Glensaw Research Farm samples. It's one of the farm sites associated with the Institute, where the soils were amended with 4 different types of treatments: the biosolids, food-derived compost, manure, and inorganic fertilizer. I've shown here the percentage recoveries obtained by spiking the soils with 2 different sizes of polyethylene microspheres and the polyester fibers. I've shown some of the pictures of the most complex soils that we have analyzed. And what we see is the hydrogen peroxide is only bleaching the non-microplastic matter. And this certainly introduces an error of margin into the analysis. And it clearly shows that we have to overcome these interferences by improving on the method. Some of the practical challenges we faced in the soils that we've analyzed so far is the time of analysis. We've got multiple flotation and filtration steps, and about 8 samples, even for an experienced analyst, can take up to a week for analysis. And 1 sample can take up to 16 filter papers. And some microplastic fibers were found to stick to the non-microplastic matter in the filter papers. So these non-microplastic matter could be organic material, plant-based, such as detritus in the filter papers. I've shown here just one slide of the preliminary results because I would like to emphasize that more work needs to be done before we can draw further conclusions. What we found was there were more fibers compared to fragments. And this has been the case in previous studies too, where more fibers were found. It's likely that the fragments, they stick to the soil aggregates and difficult to move into the liquid phase, the soil solution. That was the reasoning proposed in the paper. And you could see large variations for each given treatment. And there was no statistical difference between the 4 treatments. This was something surprising. It could be because the method needs to be improved upon, or it could be a characteristic of the land. We need to improve the method and carry out further studies to look at the concentrations of microplastics in the raw material used for treatment, in the surrounding areas of the land that was used, and incorporate land characteristics before we can make proper conclusions. Now, how can we improve the existing method? There have been some papers that have recommended apparatus such as these for improving basically the reproducibility of the method. They will not reduce the organic matter, but they help in improving the reproducibility. I think using a much wider base apparatus would be more beneficial when compared to the narrow and longer ones. This is because if we apply Stokes' law for, this is just a theoretical calculations where we assume the particles are spherical, we can see that the time of flotation increases as the particle density increases and as the particles become smaller. So this is understandable, but also as the length of the solution increases, the flotation time increases. So this will not reduce, having a reduced, but by having a wider container, importantly, what could happen is that if there is a much shorter layer of sediment at the bottom of the container, then it's much easier for especially the more polar and smaller fragments to move away from the forces of interaction with the sediments into the soil solution phase. There are also equipment such as these based on the modified flotation techniques for the separation of microplastics. So the elutrator, we have constructed an elutrator column made from stainless steel for which we could use as a volume reduction in conjunction with the density separation method. The froth flotation has also been proposed, but it has rarely been used. And most of these have been used in the marine environment, not in the soil, terrestrial soils, as far as I'm aware. So what are the ways of reducing the organic matter? One is we can try certainly the Fenton's reagent, but it can have, we need to test it because there are chances that the ion can interact with the, can get exchanged with the cation exchange sites in the organic matter that we are seeing on the slides. We can try the enzymatic hydrolysis. Recently, multiple use of enzymes have been proposed. But the problem with this is we need to consider the cost considerations and the time of analysis. In the laboratory, we are currently trying a magnetic separation method for microplastics. I cannot discuss much about it because Stuart is working on this area. And basically, we're using a high gradient electromagnetic separator for separating the microplastics as a technique in itself, or we can use it in conjunction with the soil solution to separate the organic matter from the microplastics collected on the filter paper. To summarize, there are no standard protocols yet for the separation of microplastics from the soils. And the most commonly used density separation method that we have problems with soil compositions which can be complex because of the problems with the plant-based organic matter, which can have similar densities and microscopic size. We'll conduct a time, cost, and benefit analysis to choose from the various available options that I've discussed to make improvements to the existing method. And thank you.
Thank you, Dr. Sandhya, for elaborative talk on analytical challenges of microplastics. I will hand over to Dr. Mridula to take a couple of questions from the audience, please. Dr. Mridula.
Sir, my video is off, but thank you, ma'am, for your excellent presentation. So I have very few questions from for this session. One of the question is, which layer of soil is having maximum microplastics, and what is the nature of the recovered microplastic?
Usually it's the top layer. Most papers have, most studies have used the 0 to 5 layer, or to be safer, 0 to the top to the bottom 10 layer of microplastics generally. But we don't know yet about much about, because this is an upcoming area. It's possible that the really much smaller fragments than micros, the nanoplastic range, could be going further down through the organisms present in the soil. But we don't know much about the nanoplastic size range because there's no method available as far as I'm aware. And what is the second question, Dr. Mridula? What is the second one?
Ma'am, the second question is, what is the nature of the recovered microplastic? So any specific microplastics have been observed?
I've shown in my slide that we saw one of my slides. We've not really analyzed all of the Glensaw samples, but I've seen that for the fibers for polyester, rayon, acrylic, and for the fragments, we've seen polyethylene, polypropylene, and another resin that's used in paints as a binder. Is there another question?
Okay.
Thank you, ma'am. There is another question, but not exactly related to the topic. How reliable do you think FTIR is for determining the microplastics?
FTIR gives us information. Is it compared to Raman spectroscopy? But FTIR is good for concentrations compared to Raman. Its sensitivity, I think, is a lower. It has a higher, lower sensitivity. Raman is better for really tiny microplastics, but it gives us important information about the type of microplastics. And we can automate, that will soon come up, but at the Institute, we do not have automated FTIR for the analysis of the slide, the filter paper.
Thank you, ma'am. That's all for the questions for this session. I just
Dr. Mridula, you can share the
Yes,
profile for the next speaker, Dr. Christie. Okay.
So I'll request Dr. Amita Mukherjee to introduce the next speaker.
Okay, it's my pleasure and privilege to introduce Ms. Christie Jones-Williams. She is currently a PhD candidate at British Antarctic Survey and University of Exeter. She has done her BS in Environmental Geoscience from University College of London and MSc in Marine Policy and Science from Newcastle University, Australia. She is passionate about understanding the relationship between humans and the extreme north and south of our planet. Her research focuses on investigating the possible impact of microplastics in and around Antarctic. Her PhD has taken her to the waters around Antarctic. She looks for microplastics in the surface waters there. In addition, she carries out experiments to understand the possible impact of these microplastics to the zooplankton, which form the basis of the Southern Ocean ecosystem. So she studied the presence of microplastics in the snow and built up a picture of pathways of contaminant in Antarctica. So I welcome Ms. Christie to present her work on microplastics in Antarctic. It will be entertaining.
Madam, you could share your slides.
Okie dokie. Here we go. Just going to put that.
Yeah, it's visible now.
Okay. I'm just going to change this now so that you can. Is that correct or?
Yeah. Yeah, it's visible.
You can see the full screen, not my notes.
No, we are unable to see your full screen.
We can see your notes.
Okay, you can see my notes. That's not cool. Okay, here we go. Let me change this around.
I think below there is a presentation mode. You can. There are three icons. Five icons below the slides.
Left hand side below in your screen.
There we go. That should.
Yeah.
Full screen now.
Yeah, now it's full screen.
Yeah.
Brilliant. Okay. So yeah, thank you very much. I'd firstly like to thank, of course, the organisers at VIT, in particular Professor Chandra, for this opportunity. And of course, thank you to you all for who are now online and committing this day to expanding your learning of micro and nanoplastics research. What you do at VIT is really important. It's important to expand our understanding beyond our own fields of research, because collaboration is really key to solving this environmental problem. So I'm here to talk to you specifically about plastics in all the regions and around Antarctica. As it was mentioned, I'm a PhD candidate at the University of Exeter, but I spend most of my time at the British Antarctic Survey. And I've actually spent the last few years or so going up to the Arctic, collecting marine samples and investigating them for environmental samples of microplastics. And also, predominantly, I've spent my time in the waters around Antarctica and in the sub-Antarctic. So here I look at microplastics in the surface waters and both in the zooplankton and in the water themselves. But quite recently, I came back from an interruption that I took during my PhD to lead an expedition to Antarctica itself to get my hands on some terrestrial samples where I was looking for microplastics in the snow. And this was a citizen science project as well, And I'll talk a little bit about this later on when I discuss where I think the research should be heading and where we're going. So today, I'd just like to, at the end of this 20 minutes, let's say, I'd like to reframe the context of this plastic problem. To give you a polar perspective, we'll head to one of the most remote regions of the planet, to Antarctica, my favorite continent. And my hope is that afterwards you'll be able to understand why it is that we are concerned about this very human problem in relation to a continent that actually has no permanent citizens, and why a problem that's rooted in waste needs to be explored in a seemingly pristine part of our oceans. And we'll look at this through three lenses: the past, where I will talk about what we know so far and how we got to knowing what we already know; the present, where I'll focus on the research that I do. Predominantly, I want to discuss some incubation experiment setups where we can understand how microplastics might be interacting with the zooplankton in the Southern Ocean, and then to look to the future. Where is this research heading? So we'll do a bit of time traveling first. The British Antarctic Survey, or BAS, as I'll probably refer to it for the next 20 minutes, has been studying the Antarctic marine ecosystem since the 1970s, but marine debris almost 20 years later, so in 1989. We have three monitoring stations. This is the Antarctic Peninsula here, you've got South America up here, and our monitoring stations are on the sub-Antarctic island of South Georgia, Bird Island in King Edward, and Sydney, South Orkneys. And Bird Island is appropriately named, just to give you an idea of how populated it is in terms of animal density. We have birds and seals existing in a density of 1 per 1.5 meters squared. To give you some context, to compare that to the population density of India, that's about 4 orders of magnitude greater. So we have a lot of animals here that rely upon the food and the habitat that these subantarctic islands provide. And how is this being impacted by marine debris? Well, we've had year-round sampling since 1991, and that's been macroplastic debris. Earlier, Richard Thompson talked about plastics and making sure that we were considering plastic pollution, not just in terms of microplastics, but that wider problem of macroplastic. And that's how we first became aware of plastic pollution in Antarctica, in and around Antarctica. So they were finding about 27 items of marine debris washing up on the shores of Bird Island. About 27 items per month, and most of this was actually fishing gear. When they looked at the trends, you can see that there actually isn't any real trend. There's about 10,000 items that have been recovered. But what is interesting is that 90%, over 90% of this was plastic. And this was evidentially problematic in terms of macroplastic pollution. It's hard to deny that this was a problem when you were seeing commonly entanglements of Antarctic fur seals with fishing nets around their necks. With seabirds, the wandering albatross, in fact, was associated most commonly with plastic pollution in their nests and in their stomach contents. This here on the right is a picture of some of the retrieved plastic and marine debris that was found in and around the nests of seabirds on Bird Island. But what we need to know beyond macroplastic is to understand what the impact potentially is of microplastics in and around Antarctica. And we know already that the impact of plastic increases as the size decreases ever smaller from macro to nano. To think about this, we need to think about the pathways for microplastics in the polar regions. So we can think back to the seabirds and those entanglements, and there must be plastics in the sea surface. We know that. They travel through the subsurface, through the water column, and finally settle on the seabed. But in the polar regions, we have ice as well. And this complicates things because ice can act as both a sink of plastics and eventually, when it melts, a source of plastics. But again, this is far too linear. We need to understand what interaction the microplastics within the water column and through the water system have with the ecosystem. So how do they interact with the Southern Ocean ecosystem? Well, it's comprised of keystone species at the very base of the food chain. And this means that just a few species are of paramount importance to the health of organisms across all levels. So from small fish to whales, seabirds, and seals. And for the rest of the talk, I'll focus on small pelagic crustaceans. So that's predominantly the Antarctic krill, carnivorous zooplankton. I'll refer predominantly to pelagic amphipods, so those amphipods that exist in the water column, and then other herbivorous zooplankton, which we've done a fair amount of experiments on to understand the interaction of microplastics with zooplankton, such as copepod, might be more familiar. So bearing this ecosystem in mind, and looking specifically at the surface waters, we wanted to know whether microplastics were interacting with these zooplankton, because these guys are what feed upon microplastics potentially. These microplastics exist in the same space and at the same scale as the diatoms and the microzooplankton that they feed upon. So they are our gateway to the ecosystem, if you like. And in 2017, Waller et al. Reviewed what was really only a handful of peer-reviewed papers looking at microplastics around Antarctica, and most of these here, so the green crosses represent microplastic studies, but actually they were predominantly just citizen science projects, and very few peer-reviewed papers had been brought out by 2017. And there was a lot more incidences, reportings of macroplastics. Here are a couple of examples of some studies that were done up to that point, and they were focusing on the deep sea sediments. And then we've had a few studies since then which have looked at microplastics in the surface waters around Antarctica. But again, this is a handful of studies, considering the Southern Ocean is 22 million kilometers squared, or 71.8 million kilometers cubed. 3 studies is not enough to infer a threat perhaps from microplastics, or a real understanding of what the concentrations are. We need 2 main things in 2017: that we needed more environmental samples to get a more accurate coverage of pollution, and that we need to understand how this pollution interacted with the ecosystem. So the possible impact of microplastics on the Southern Ocean ecosystem, we need to have a look at that impact on zooplankton, which is where microplastic is bioavailable. So we could look at other experiments that had been done. One of my professors, Dr. Matt Cole, has done a lot of experiments on the Calanus copepod, and this copepod exists in oceans worldwide, to understand really what the impact of microplastics was on the energy source that a copepod has access to. So here we have our Calanus copepod. The blue fluorescence particles that you can see there are actually microplastics which have been ingested during an experiment. The control shows the amount of micrograms of carbon which were taken up by the copepod from its phytoplankton food source, and the grey bar shows how much carbon is taken up from that food source when it's in and amongst a pool of microplastics. It basically shows that microplastics are essentially a waste of energy. And when we think about that energy that's used by any zooplankton, we can see that energy is consumed and lost through egestion. So on the left there, you have a fecal pellet with those blue microplastic particles in from that very same experiment, and through metabolism. And again, the remaining energy reserves are basically used for survivability and reproduction. So what happens then when we add microplastics to this energy resource? Well, of course, what we see is we reduce the available energy to the copepod to survive. More energy is taken up for removal of this microplastic and to survive itself, so increasing that metabolic rate. And then the overall energy reserves that are left for that organism reduces their overall survivability and the health of their eggs. So we knew this going into looking at microplastics around Antarctica, but we need to consider, well, what are the things that make the Southern Ocean ecosystem special? So we have animals which rely on very specific requirements for food and a specific temperature niche, which are inextricably linked to the dynamics of ice accretion and ice melt. So we know that they're going to be impacted by global changes, global warming. And secondly, these organisms have lower genetic differentiation due to existing in this extreme and therefore geographically isolated part of the world. And with this, they have slower growth rates as well. So we have microplastics being perhaps more problematic in a region such as the extreme south, because they are adapted so acutely to this very specific environment. And even just a small amount of this pollutant potentially could have a bigger problem than you might anticipate in more temperate regions where organisms are more used to heavily polluted coastlines. So we wanted to know, okay, well, do Southern Ocean zooplankton actually interact with microplastics? Well, in an experiment, in an incubation experiment, we know that they can actually fragment microplastics down. There was a really cool study done by Dawson et al. In 2018, and it showed that they actually contribute to that fragmentation of microplastics, nanoplastics, by chomping through and ingesting smaller particles when in an incubation setup. And so taking that keystone species, the Antarctic krill, we cannot simply imagine, though, microplastics in the Southern Ocean as one single stressor. That's it, that's all it has to cope with. No, we've got to imagine the suite of stressors that already exist. So we need to know how microplastics combine with those other stressors to impact krill health, but also how do these pre-existing stressors affect the ability of the krill to combat this new stressor? And I keep saying stressor, but what are we talking about then? We're talking about tourism, the increase in shipping around Antarctica. You can see on this map here, there was a study done just last year that assimilated all of this data of shipping traffic around Antarctica, and it found the Western Antarctic Peninsula is here to be a hotspot of traffic, and particularly increasingly so. Incidentally, this area around the Western Antarctic Peninsula and what we call the Scotia Sea here is also where we find the highest densities of Antarctic krill. So related to that as well, increasing shipping is also coming from fishing. And then, of course, we have all of those stresses related to global warming, so ocean acidification, increased temperatures, increased ice melt, and then as a result of all of these complications, migrations of Antarctic krill poleward to the cooler regions as the subantarctic warms up. And then we have also invasive species which come along because we have this changing environment, and then other pollutants such as persistent organic pollutants and heavy metals. And we know as well that not just being a pool of stresses, they are also increasing, and the rates of these are increasing as well. So what does this current research tell us? Well, it tells us that there are two lines of inquiry that we need to follow to assess risk: to determine exposure through environmental sampling and to assess the impact through incubation experiments. So to determine exposure through environmental sampling, we recently released a paper in Environment International, and I would urge you next month to have a look at Environmental International. There's a polar plastic special issue which has a huge suite of studies that have been published just this year on microplastics, macro and nanoplastic pollution around the continent. Really, really important, really cool stuff. So we were looking at microplastics in the surface waters, but also in the zooplankton in those surface waters. So here we've got our yellow diamonds where we looked for microplastics in the water and in the zooplankton, and the yellow triangles where we just looked for microplastics in the zooplankton. Red are some stations where we collected some zooplankton, but we didn't look for microplastics there. We used this net, which is a Newston net. We trawl it parallel along the surface. We could filter through about hundreds of thousands of litres worth of water, but that's because we used a pore size of about 300 micron. We've now developed a net which is actually smaller with its smaller pore size, which means we're not missing that vital size fraction. But at the time, this is what we had, and this is what standard practice was, but that is evolving. So just some brief findings that we had. We found that the methods to identify sampling bias are crucial. So you could pick up a sample, you could have a look at it and go, hey, there's a lot of microplastics in here, but actually you're taking that from the ship. So you need to be really, really careful with making sure that what you're measuring is something from the environment and not from just being there. So we used the Fourier transform infrared methodology to basically build up a contamination library so that we could realize what it was that was coming from the ship itself and remove that from our final concentration. And what we found was 46% of those kind of optically identified microplastics were in fact from the ship and from things that were out on the deck. It's really important before you make any assumption on what the concentrations are that you have good polymer analysis to understand what polymers they are and where potentially they're coming from. Secondly, we found that the microplastics concentrations are actually really low. So we found 0.013 particles per meters cubed. That's comparable to other studies which happened recently as well around the same area along this peninsula. When we scale that up to particles per kilometre squared, which is often used when we look at sort of global concentrations, it was 1 order of magnitude higher than what was been found by a circumnavigation study in the same year around Antarctica. But overall, it was 1 order of magnitude less than the global average. So still not much, but significant because it's there. And we know because of that very specific niche environment that we have, that it's significant because these animals are not prepared for this kind of stressor. So we also wanted to look at not just the microplastics that were in the water around it, but also the microplastics that were in the amphipods samples themselves. So often when people look at the surface waters, they define this potential encounter rate, which is the amount of microplastics they find in the water divided by the amount of zooplankton they find in the water. But we also wanted to add this observed encounter rate to see what it was we were finding in the zooplankton and compare that to the amount in the water itself. And we found that our concentrations, the observed encounter rate was higher than what you would expect to find looking at just the surface waters themselves. So it goes to show that we need to be looking beyond just the water, but we need to be looking inside those zooplankton. And then very briefly, and I'm happy to talk about this a little bit later, if there's any questions regarding—
We have another 2 minutes to go.
Okay, but we did find lots of different polymers, but predominantly they were coming from paint resins and polyethylene, the most common plastic pollutant that we have. To address the impact, so ecotoxicological, this is what I'll finish on, and this is really important. It's to make sure that you're designing multi-stressor experiments. So here, this was a paper that was released a couple of years ago, looking at mercury levels in Antarctic krill. We know very little about the mercury cycle in the fact that it's very complex, but we do know that it exists both naturally and through anthropogenic sources in our environment. So I was interested to know that because Antarctic krill have high concentrations of mercury in them, and we know that mercury comes from waste incineration, so part of that same sort of problem with poor mismanagement of waste. We know that mercury bioaccumulates in these Antarctic krill. How then, if mercury can scavenge onto particulate, can it do the same on plastics, and therefore can it interact with krill in this fairly interesting way that perhaps we haven't considered before? So rather than just looking at microplastics ingestion with krill, I wanted to look at how that mercury interacted with the microplastic. So my hypothesis was that mercury scavenging of microplastics will reduce the uptake of mercury by Antarctic krill. So I set up an experiment on the ship, and on the ship, you're in a moving lab. So you lash everything down, you put it under benches, you make it safe. And then we designed an experiment that basically took a control of krill in Kilner jars. We had a Kilner jar of krill with mercury. And then we looked at 2 different types of polymers, so polyethylene and polystyrene, which is commonly used to look at multi-stressor experiments with microplastics. And then we did combined assays of polyethylene, polystyrene, and mercury. I've been told today that I can get back in the lab, so I will have results for this soon, but I haven't been able to analyze the samples. But this is just to show microplastic uptake by those fluorescent particles in the krill. So just to finalize, where is this research heading? Well, we've talked about that we need to carry on with determining exposure through environmental samples and to assess impact through ecotoxicological experiments. But we also, as a part of that, need to understand what it is that we need to focus on, what we need to prioritize. There's very little information about microfibers. We looked at microfibers and ours, but I wouldn't say that we use the correct methodology to determine good concentrations. We need better temporal coverage to understand how ice melt perhaps is influencing the concentrations of microplastics. We need better spatial coverage. We can't just concentrate in this one section around the peninsula, but perhaps we do need to prioritize other hotspots of zooplankton densities. And then we need to look not just in the waters, but also the Antarctic continent. There's no number yet for that. And there's no standardized methodology. This is something that's developing. And to assess that impact, I urge anyone that wants to set up an incubation experiment with microplastics to look at one of those other individual stresses to see how it might interact with it. I think that's really, really important. And on top of all of that, we need to educate a really great thing at the moment is that we have momentum, we have public interest in plastic research. And when you talk about how plastics might influence an environment so remote, so far removed from our home, and that our behaviors back home might have an influence on this incredible environment, then it's really important that we get people to realize why the Antarctic environment and how the Antarctic environment might be impacted by plastic. So I'd urge you to have a look at the Antarctic sabbatical online. There'll be results coming out soon. And that's just an example of a citizen science project where we collected microplastic samples in the snow. And we aim to disseminate the information via the 5 volunteers that came with us. And that was something that was organized through Airbnb. And just to finish off then, I'd like to conclude. So to determine exposure, to educate and to assess impact, we found from our studies that there are low concentrations of microplastics, but these are still pertinent in places like the sub-Antarctic and Antarctic. Method development is key to identify this real contamination. So thinking about FTIR again, focus on those zooplankton hotspots. And if you're going to do experiments, make sure that they're multi-stressor so we can help build up a better understanding of true impact. I'd just like to say thank you very much again to everyone, and thank you for listening.
Thank you, Dr. Christie. You had been working in a very hostile environment. I have one question from the participants. What is the effect of psychrophiles for conversion of plastics to microplastics?
Of what, sorry? Of?
In Antarctica, what is the effect of psychrophiles? That is, what is the effect of bacteria or microorganisms on conversion of plastics to microplastics?
That's a really interesting area of research, and it's not one that I'm personally involved in, but I certainly believe that because we have these, we're talking about very different temperature ranges, that actually you might anticipate that the bacterial influence in a cold part of the ocean, such as Antarctica, is less than you might be finding perhaps in more warmer temperate parts, or in fact just very different. So I think that's something that's not being researched at our institute, but it's potentially being done elsewhere. There's the Scientific Committee on Antarctic Research, and they have a plastics working group, and they will potentially have somebody within that area that's looking at bacterial influences. So I will, if that person would like me to pursue further, I can certainly ask, but I haven't considered it myself.
Thank you, ma'am. Can I invite any questions from the panelists?
Ms. Christie, I have a question. Why you have selected mercury as a co-stressor for your work, or what are the other co-stressors which may be possible in Arctic environment?
Sorry, could you repeat? I've got really quite terrible speakers on my laptop. Could you just repeat the question?
Why you have selected mercury as a co-stressor for your work, or there are possibility of other co-stressors along with the microplastics in the environment, in the Arctic environment?
I'm sorry, I can't hear.
Okay, it's not a problem. I can share the question later with you.
Yes, please do. You've got my email address, so please do send me.
I'll do that.
Thank you.
Okay, thank you.
Thank you, Dr. Christie. We'll move ahead to the next distinguished speakers. Over to Dr. Amita Mukherjee to introduce the speakers.
My pleasure again to introduce Dr. David. He has done BSc in, can you go back to previous slide? Yeah. He has done BSc in physics and MSc in laser physics and PhD in applied physics from RFCS 7 Cranfield. He has been a postdoc in University of Manchester from 1993 to 97, adjunct professor in GIT from December 2015 onwards. He is a visiting professor to Institute of Electronics, Microelectronics and Nanotechnology, France from 2008 onward. He has been a lecturer at University of Plymouth from 1997 to present. His current research focuses on sensor development for characterization and optimization of materials and devices. His research also focused on two-dimensional materials such as graphene and derivatives, along with thin films and solar cells, and nanoporous aerogels for water treatment. He focuses on the surface plasmon resonance for detecting ultra-low concentration of nanoparticles in water. He will be joined by his student, Mr. Jonathan. Can you please show the next slide? I'll introduce Mr. Jonathan also. He is an interdisciplinary scientist currently studying for PhD at University of Plymouth. His work focuses on water filtration using biopolymer aerogels. He has got his master's degree in robotics, producing graphene-based piezoelectric sensors using thin films. His research is focused on using graphene oxide for filtration purposes. He has been awarded the first prize for a poster at the first International Aerogel Conference at Newcastle University in the UK, and selected for Junior Research Exchange Fellowship to collaborate with IISc in Bangalore, funded by Indo-UK Water Council. I welcome you both. Please share your views with us.
Okay.
Thank you.
Try the share screen.
You can share the slides, please.
You can share your slides, sir.
Yeah, I'm trying to. I want to have this in. So you can see my main screen.
Yeah, looking good, David.
Yeah. Okay. I say so. Good afternoon, almost good evening. So this is a talk that Johnny and I are doing together. We've been working for quite a while now on contaminants in water, from nanoparticles originally through to now. A focus has been on heavy metals and pathogens. So we've been working on sensors and filtration materials to kind of achieve good quality, safe drinking water for sort of like rural Indian communities. And this has kind of led us into this particular area that so we got some interest from the Impact Lab that kind of ventured through Plymouth and Exeter, where they were interested in sort of filters for sort of domestic washing machines. We've also started to make some discussions in this area as well. And we kind of thought, okay, microplastics is one thing. There are people developing some systems, but we kind of think we want to focus on the nanoplastics. So this talk will kind of explain why nanoplastics. We've heard a lot of talks in the last sort of couple of hours that kind of show exactly why nanoplastics are an important thing to be aware of. And we see, you know, they've been disruptive in the sort of on the human level. And then we're going to look at sort of essentially how can we use the sort of sensors that we are familiar with in our current research to design and characterize solutions to actually achieve filtration of nanoplastics. So we were looking at a system which would fit in beyond the current designs of microplastics. So these filters are essentially designed to work with water which is essentially clean, but to remove specific contaminants. Okay, so these nanomaterials, they are sort of very kind of buzzy. People talk about nanotechnology in all the positive things, and there are so many positives. But we know that when these particles become of these sort of size, sort of sub-100 nanometers or so, then we know that they are disruptive on the cellular level. And we know that it can have many effects. For example, we looked at some studies on sort of filter feeders, but we know that these kind of filter feeders also move up through the food chain. So that the problem is it goes far beyond even this kind of basic understanding of what may be going on. And but this work is not totally new to us. So one of my colleagues, Richard Handy, who we work with, so we've been working. Say we've done a lot of work on detecting silver nanoparticles. Again, it's a toxic particulate for aquatic species, and so we know, for example, some studies that we've done in Plymouth, you can see here what actually happens where, say, a trout is exposed to titanium dioxide nanoparticles. Now, we do know that the way these particles leach out means that the levels in the environmental water are relatively low. But when you've got 800,000 microplastic particles, fibers leaching out into in a domestic wash, how many other particles have we got that have been missed? So we could be talking over 1 million when we include the nanoplastics. And so this is our kind of focus on our work. And it also kind of brings into this other kind of arena about this kind of recycling. This came up, Richard Thompson, recycling plastics. A brilliant solution in many ways, but not everything is a positive. So the thing that we've been interested in is in the sort of polyester fabrics that come about from people recycling PET. So we often talk about the nanoplastics composed of rPET. There may be others. I know the cosmetics are an issue. In this case, it may be less of an issue, but obviously cosmetics leach into sort of fabrics as well, and they'll also be part of this kind of process. So we're interested to see if we can detect these nanoplastics that are post microplastic filtration. So we want to know what is in there and what kind of size are they. Now everybody seems to be using FTIR for microplastics, and yeah, FTIR is an extremely useful tool. It's gives us the means of exploiting the infrared signature of materials, so we can easily identify them. But once you go down into the sort of like especially the smaller scale of nanoparticles, nanoplastics, then things become a little bit more challenging, because they then go beyond the kind of diffraction limited detection that we can get in conventional optical systems. So even Raman struggles in this area, but not all is lost, because this kind of works in an area that we're also interested in for some of our other work, where we're developing the same kind of sensor technologies to look at very low concentrations of biomarkers for diseases. And what you can see here is that that when you look at the very small Raman spectra from sort of particles which are down in the sort of like a few tens of nanometers, then the signature that you see is very weak, because it's dominated. It's a very low signal you get from a Raman. It's only about 1 in 10 to the 8 of the actual scattered signal that you would get. So it's a very small signal. So what is the answer? The answer is probably a combination of sensors, but one that we're going to be focusing is on a surface enhanced Raman spectroscopy, which kind of uses our expertise in plasmonics to develop sensor chips which can detect down to a few picograms per milliliter. And as you can see, the sensitivity is pretty good compared to what you get with normal Raman. You have to look at the numbers on those scale bars. So we know this is one of our tools. We're also interested to combine our other sensors. And I know TEM is a really good way of looking at particle size, but it's more of an elaborate process, maybe not quite the way to go, but something we won't exclude. So we may find that we may use a nanosite tracking analysis, which is a much simpler to use system, which produces very comparable data to TEM. So this may be one of our solutions. In order to, so this will tell us size and shape. Then SIRS will be our means of determining kind of like the composition of these materials. But we also have some other ways we may be able to look at real-time monitoring using some microfluidic systems adapted from work from our colleagues in IISc Bangalore. And I'll soon move on to be able to pass over to Johnny. So you can see this is the kind of system that we've been looking at based around the recycling of things, and everything goes very well as you go round to here, to here, to here. And then it's when you get to this particular point here where the garments are produced from these materials, and what we know that they then pass through and into the rivers and the sea. So this is obviously a problem for these filter feeders, aquatic species, and it's unknown how many of these will actually get into the domestic water supply. And so the kind of what we're looking to do is to go into a situation like this where we can incorporate one of an aerogel filter designed specifically for these plastics. So I'm able to pass you over to Johnny, who will kind of go through the work that he's been doing to develop these aerogels.
Thank you, Dr. David. That's great. Thanks for everyone for being here today. So yeah, so we're looking at filtration of microplastics, but also nanoplastics. And if we take a little look at this next graphic here, we'll see the kind of range of solutions that you have when you're working in the nano scale to remove any object or ion. Traditionally, you've just relied on nano filtration would be your go-to place for a small piece of plastic, but we've also very familiar with using reverse osmosis, especially in the treatment of removing salts, dissolved salts from water, and also toxic metals. We spend a lot of time working on toxic metals, and I'll show you how toxic metals overlap into our interest in nanoplastics, and how we can potentially use the same systems and the same filtration systems for removing these contaminants. So we've heard earlier on that during the wastewater treatment process you use something like activated carbon to remove toxic metals, and at the same time this activated carbon is also collecting some of your micro and nanoplastics, and this is then forming a sludge which is then used for fertilizer on soil, soil conditioning, etc. So we're taking this very efficient wastewater treatment process that can remove up to 95% of the microplastics from the water supply, but then we're depositing it onto the soil, and then we've seen lots of very interesting presentations about how this then persists in the soil and what happens to it and how it degrades, etc. So we really want to try to avoid getting to this place, and we've talked about how to sort of avoid putting it into the environment in the first place. So our kind of our solution, which is very similar to using activated carbon for toxic metal adsorption, is to use a different form of carbon, is to use graphene oxide in an aerogel form. Now, you're wondering what aerogels are. They're low density solids which have a porous network that can allow a nice, a reasonable flux rate of water to flow through the network, but also many active binding sites for toxic metals. And we can see that this is also an aspect that can be utilized for nanoplastic removal due to the surface charges of these very small. As the particles get smaller, we all know that the surface charges play a much more important role. So we've developed aerogels here for toxic metals, and so the simple process that we've used for aerogel production, I'll give you a quick overview, is that we're making a precursor solution for aerogels. This is the fabrication steps that we use. And this goes through like a hydrolysis and condensation reactions, forming sort of nucleated nodes where you form a gel structure. This is then processed, and we use a freeze-dry technique to produce our final aerogel. And these have been proven to be very effective in removing high concentrations of toxic metals. Not only do they remove toxic metals, but they also can perform oil-water separation, which we'll come to in a little bit in relation to nanoplastics. Dye absorption, you can see here a nice image of methylene blue dye being absorbed by aerogel structures. So they're very, I would say they have a wide range of applications, very versatile. So particularly nanoplastic and microplastic adsorption into aerogels. We've been working on this principle that there is a charge surface inside our aerogel structure, usually negatively charged, and we've historically adsorbed positively charged cation, lead ions to the surface of the structure, and due to its surface area, it's incredibly efficient and can absorb huge amounts without reducing flux rates. So this is a really important distinction between a single thin filmed layer nano filtration system where clogging and reduction in flux rate occurs, especially with the presence of natural organic matter. So we've got this, we've got the structure and the functionality all built in together. It's a very robust network you can build with biopolymer aerogels, and we've made these particular ones totally biodegradable and biocompatible to not produce another tertiary nanoplastic in the removal of your plastics in the first place. So this is some of the data that we have for our lead absorption, and we can see that we're removing very high levels of lead from the environment. So these aerogels are very, very efficient, and they could be potentially used for nanoplastic removal. Now, our first concept, this early stages for removing nanoplastics is to really mix the nanoplastic with some form of oil. Now we've used rapeseed here, but we'll probably use a slightly denser oil.
Jonathan, you have another 2 minutes to go.
Thank you. And we could, what we propose is to mix the nanoplastic with the oil in a, as a sort of a two-stage filtration step. And as you can see here, these aerogels are very good for oil-water separation. So with the lithophilic characteristics of the plastics, they will then remain inside the oil. They're hydrophobic, so they'll be separated out from the water. And with this very neat and functionalized aerogel surface, you can see the oil is sitting at the top there, and the water is filtering through. So this is a hydrophilic and oleophobic 3D structure. It will. Our kind of instinct is that it would make sure that the nanoplastics are suspended within the oil, and then you have a nice clean water, solution afterwards. So this is one of our concepts, and we think that this could be utilized for nanoplastics. Also, we've been working also with highly conductive aerogels. So if we're working on a charge principle, which we have been using our absorption for lead cations, and if we're getting very, very small nanoplastic particles that would be potentially adsorbed to the surface via its surface charge, then we would be looking to use something like this, which is a kind of capacitive deionization system, but then transferred into the separation of nanoplastics by sort of partition of charge, really. So and this could be implemented in a low cost environment that's very low power, and the benefits of using this is that you could then wash out your structure, and then you could then reclaim your plastics and recycle them properly, and then drink your water. So that's our two proposals for cleaning up nanoplastics here at Plymouth, and we're in the early stages of working on these things. So as David said, we've got disruption of cell walls. We really want to prevent primary release in the first place of any plastics, especially nanoplastics. These aerogels, I think, can work for separating nanoplastics from water via the two-stage process of oil and water separation, or by a charge-based capacitive deionization system. And then we talked a little bit about, me and David, if you can come in there, David, that you know, to wear some sort of natural biodegradable clothing, so we're not washing this constantly into the environment. I mentioned cotton, and then David says there's a lot of water used in cotton, so we have to preserve our water too. It's a balance of you know, of risk here. So yeah, so that's how it works so far at Plymouth, and we're very pleased to present some of our ideas to the audience here and everyone at BIT. So I'd like to thank you very much for listening to our presentation. Thank you.
Thank you, Dr. David and Dr. Jonathan, for a very informative speech. I have a question for you. So how effective do you think affinity chromatography will be for separation of nanoplastics?
I think that's one for you, Johnny.
Affinity chromatography. Yeah. I don't think it has anything to do with my talk, and so I can't really answer.
No, it's something we've not explored. I
would honestly say I knew nothing about it. But thank you for the question. Please, another question would be.
Yeah, we just we probably come along this path based around this kind of so far the kind of analytical tools that we use so far, but we have used far more. We've not gone into chromatography. We've gone into ICP-MS. But we're trying to find ways that we can do things in a simpler way, so it's easier to develop and characterize materials. This is why I'm not so keen on TEM, because it's a big technology to apply. Whereas a nanosite, we can go into one of the labs and drop a sample into it and see the answers in a few minutes without any high vacuum, etc. So we're trying to do what we can to simplify the process. That's why SURS is another good tool, because we can apply it to a standard Raman. Developing the chips is difficult, but we're working on that with colleagues in Imperial as well. So we're trying to come up with all our water working in it. It's all been about affordable, low-cost technology that can be used robustly and in environmentally friendly ways. So we're always keen to explore some new avenues. So now with Amitabha and Sina, we've already started discussing some collaborations in this area as well. So it's quite exciting to see a new kind of direction with our work.
Thank you, Dr. David. I thank all the distinguished speakers who have been present with us for this session. I request all the participants to join the next session at 5:30. Thank you, sir.
Thanks very much. Thanks for your time.
Yeah, thank you.